Good day everyone, we are Group 4 and today we're going to be presenting on complement deficiencies and the susceptibility to bacterial infections. The members of our group are Aruna, Ayana, Kadina, Nyala, and Sophia. We hope you learn a lot and enjoy this presentation, so let's get started.
Our first slide is a brief introduction to the complement system and its importance. We'd like for you to keep in mind the aim of the complement activation, which is to eradicate pathogens or to limit the bacterial infections, as well as reduce its dose to manageable levels and initiate recovery.
In the classical pathway, as seen in the diagram, the binding of C1 to the antibody complex attracts C4, which is then cleaved by C1R and C1S into C4A and C4B. C2 is attracted by C4B, which is then cleaved into C2A and C2B. C4B and C2A form C3 convertase, which then cleaves C3 into C3A and C3B. C3B binds to the C3 convertase, which ends in the conversion of C5 convertase. C5 forms C5A and C5B, which undergoes further reactions to produce the MAC complex. In the alternative pathway, an antigen-antibody complex is not required and occurs when a foreign cell surface material enters the host body. C3 complement proteins interact with the antigen and then undergoes hydrolysis to produce C3A and C3B. C3b binds to the intrusive cell, which causes a series of reactions that results in the formation of C3 convertase. C3b binds to C3 convertase, producing C5 convertase, and the process continues similar to the classical pathway. The leptin pathway is activated when leptin attaches to mannose-containing structures on the surface of the microbes. The system is similar to the classical pathway as it utilizes the cleavage of C2 and C4 complements. However, the proteases C1R and C1S are replaced by MSP1 and MSP2. The rest of the pathway continues the same as the classical pathway. Next, we have Table 1, which lists the various proteins involved in the classical pathway of the complement activation and their functions. In Table 2, we have a list of the various proteins involved in the alternative pathway of the complement activation with their functions. And in Table 3, we have a list of the proteins involved in the lectin pathway of the complement activation and their functions. Table 4 shows a list of various proteins involved in the terminal pathway of complement activation and their functions.
Introduction to complement deficiencies. A complement deficiency is an insufficient amount or the complete absence of complement or regulatory proteins in the blood. This deficiency often is associated with increased susceptibility to bacterial infection. Additionally, there are many autoimmune diseases associated with complement deficiencies. One example is systemic lupus erythematosus. This is a serious condition that leads to inflammation, tissue damage, and organ damage. A deficiency of complement proteins or regulatory proteins often affect the activation of pathways mentioned earlier. Since these zymogens follow a cascade, if one of the proteins is deficient, the entire pathway is affected and cannot be activated. This puts the host organism at risk for bacterial infections such as meningitis and pneumonia, as there is a lowered immune response. Risk factors. These deficiencies can either be inherited genetically or can be acquired from a previous infection. Some of the main risk factors include age, sex, and race. There are certain forms of complement deficiencies that may occur in the earliest stages of life, while some may occur in the latest stages of childhood or adolescence. Whereas properdin deficiency is an X-linked trait that is only present in males. Certain races are more prone to specific deficiencies. For example, properdin and C2 are more prevalent in Caucasians. Now we're going to be discussing complement deficiencies that specifically increase the susceptibility to bacterial infection. The first components we have are C1, C2, and C4. These components are found early on in the classical pathway. If any of these components are deficient, they cannot activate the classical pathway. Thus, there is an increase in pyogenic bacterial infection. The next component is C3. C3 is a very important molecule as it is the convergence point of all of the pathways. It is also very important in the production of opsonins, which assist in the destruction of pathogens. C3 deficiencies can also lead to pyogenic bacterial infections. Next, we have C5, C6, C7, C8, and C9. This is responsible for creating the membrane attack complex, also known as MAC. A deficiency in any of these proteins will prevent the MAC from being formed. This deficiency leads to Neisseria infections. A deficiency of mannose binding lectin prevents the lectin pathway from being activated, and this can lead to recurrent bacterial infections. Lastly, a deficiency of properdin leads to the inability to activate the alternative pathway, and this can lead to very severe bacterial infections.
Here's a review of what we discussed so far. Figure 2 illustrates the convergence of the three pathways to the terminal pathway. Let us now explore how the complement system is activated in response to a bacterial infection, provided that there are no complement deficiencies. Complement proteins are present in the blood and body fluids as inactive precursors or zymogens. The complement cascade becomes activated upon contact with bacteria. An activated complement cascade on the bacterial surface triggers a variety of responses that helps to limit, clear, or reduce the dose of the bacteria to manageable levels. Figure 3 illustrates how complement activation results in the formation of the membrane attack complex MAC, seen in Figure 3a, and how the complement labels the bacteria with C3-derived products to stimulate engulfment of bacteria by phagocytes, as well as trigger the development of an adaptive immune response, illustrated by Figure 3b and 3c. Let us now discuss this figure in detail. The main effector functions of complement are driven by the cleavage of two central complement proteins, that is, C3 and C5. Firstly, the complement cascade is triggered by the recognition of bacteria via soluble pattern recognition molecules or antibodies that bind both gram-positive and gram-negative bacteria. The C3 convertase formed on the bacterial surface cleaves the C3 protein. This generates C3b that exposes a reactive thioester bond. This is important as it allows C3b to covalently attach to hydroxyl groups of carbohydrates on the surface of the bacteria. Deposition of C3b on the bacteria's surface will induce and enable phagocytosis by opsonization, leading to direct killing of the bacteria. C3b and its breakdown product, inactive C3b, are recognized by complement receptors on myeloid and Kupffer cells. The labeling of the bacterial cells with C3b and inactive C3b also stimulates an adaptive immune response by directing the transport of the bacteria through lymphoid organs and by enhancing antigen presentation to adaptive immune cells. This will allow the development of antigen-specific recognition receptors that will allow for specific recognition and elimination of bacteria, as well as support immune responses by forming memory against reinfection. Deposition of C3b on the surface of the bacteria also alters the specificity of the C3 convertase. If the concentration of C3b is high, the C3 convertase changes into C5 convertase by switching from the C3 substrate to C5. Among activation of C5 releases C5a, which is a strong chemoattractant that aids in the recruitment of phagocytes towards the site of infection and induces apoptosis. C5a also triggers basophils and mast cells to produce histamine, which induces inflammation and vasodilation. The inflammatory function of complement activation is important to trap the bacteria, localize the bacterial infection, and prevent its spread. The parallel formation of C5b also triggers the assembly of MAC. This specifically kills the gram-negative bacteria. Gram-positive bacteria is protected against MAC-dependent killing due to their thick peptidoglycan outer layer that the MAC, which prevents the MAC from inserting into its cell membrane to form perforations that will result in the bacterial cell death. So, to tie things together, the use of the complement system to kill bacteria is the most effective way to kill an invading bacteria. While both the labeling with C3b and the MAC-dependent killing of gram-negative bacteria occurs within minutes, phagocyte attraction and subsequent intracellular killing takes longer, approximately 30 minutes to 1 hour. The complement system is extremely important to clear bacterial infections in patients with genetic complement deficiencies who are prone to recurrent infections, as well as provide defense against bacteria because some bacteria are resistant to various steps in the complement cascade. This slide gives a short description of Figure 3. These are some of the reasons why the complement system should be regulated. These are some of the ways the complement system is kept in check.
Even though some complement deficiencies may be asymptomatic, others can lead to serious symptoms and other harmful effects on the body, such as these mentioned on the slide. In Figure 4, we see 6 individuals experiencing different versions of cutaneous vasculitis due to a deficiency or mutation in the C3 component. When presented with symptoms, clinical tests are required in order to properly diagnose these individuals with the deficiency. There are multiple tests that can be performed, a lot of which are blood tests. Primary testing is done to determine the activity of the pathway. A CH50 assay is performed to determine the activity of the classical pathway. An AH50 assay is performed to determine the activity of the alternative pathway. A mannose binding lectin test is done to determine the activity of the MB lectin pathway. Secondary testing, on the other hand, determines the deficient component by directly measuring protein concentrations or by testing the functionality of the protein. Genetic testing is done to determine the deficiency of a particular component or regulatory factor in terms of hereditary deficiencies. For persons who are asymptomatic, a complete blood count can be done to determine the level of white blood cells, which serves as an indicator for infections. Sometimes these blood tests must be repeated to ensure accurate results.
When diagnosed with a complement deficiency, standard therapy is usually the first form of treatment applied. It involves early detection of the infection followed by treatment with antimicrobial therapy. Some deficiencies often result in recurrent infections, so patients often benefit from antimicrobial prophylaxis. Complement deficiencies are treated as immune deficiencies, hence immunization is received for illnesses that the patient is more likely susceptible to. The antibody response is also monitored following vaccination, since there is an inability to activate complement that impairs the immune response to a particular extent. Complement protein replacement is a specialized form of treatment. For example, C1 inhibitor deficiency is treated with protein replacement. However, complement proteins possess rapid turnover, can trigger immune responses where autoimmune disorders would require more immunosuppressive therapy. Lastly, we've provided all the key points made in this presentation about the complement system and complement deficiencies. Here's a list of references we used. Thank you all for your undivided attention. We hope we are able to teach you something new today. The floor is now open for questions.